That's great. Hi, I'm Adam Scott, a Bayer Science Fellow and lead data scientist here in breeding of crop science. It's my pleasure to introduce a good friend of mine, Kuan-lin Huang. Born and raised in Taiwan, Kuan is now an assistant professor of genetics and genomic sciences and PI of the Computational Omics Lab at the Icahn School of Medicine at Mount Sinai. After being the only graduate to earn 2 honors, molecular biology and studio art, at Wesleyan University, he completed his PhD in statistical genomics and then a postdoc in multi-omic software development at Washington University in St. Louis. He has authored over 60 peer-reviewed publications, including lead authorship in the top scientific journals, and is the author of the bestseller Solve It Yourself. Kuan was a co-founder and CEO of the startup DeepGene in grad school and currently serves as the co-founder and chief unboxer of openboxscience.org. A good day of his life typically involves great people, hikes, books, dark chocolate, bubble tea, surfing, and tennis, and a 20-minute nap. Welcome, Kuan.
Thank you so much, Adam, for the introduction. It's great to be back in St. Louis as I did my training here. This is showing the view of Manhattan, a very different place. If you ever come, the building in front are mostly from Mount Sinai, Upper East Side, and next to the Central Park, really pretty in the fall when the leaves change color. So if any of you come there, the tennis court is just right next to Sinai. Cool, go ahead. Next slide. So today I'll talk about 3 brief vignettes of our research-related tasks, mostly in how genomics AI may shape precision medicine. And first, I will talk about a small research project that we start to generalize prediction from 1 to 2 tasks. Second is something that we're currently working on to have bigger foundation model that solve a lot of tasks potentially and can be fine-tuned for specific purpose. And lastly, perhaps the single task to solve it all to come up with agentic LLMs that can automate a lot of what we do. Next slide. Before we go into genomics, I'd like to talk about the current state of precision medicine. And this is true in a lot of cancer care currently, where we have this classical model of cancer origin. In a given cell, you might have a lot of mutations, and through sequencing, you can discover the genes that have mutations. And some of these genes can be thought of as breaks in the cell that control chromatin integrity or DNA damage repair. And those breaks you want to keep on. Whereas on the other side, you have the gas pedals that are activated for cell proliferation during development. Next slide. And so in cancer, what we found is a lot of these breaks got broken and a lot of gas pedal get pressed on. So your cells move from normal to disease state. Next slide. And that eventually give rise to uncontrolled cell proliferation and lack of DNA repair that keep acquiring mutation that lead to disease. Next slide. And so what we have now in precision medicine that has come true in cancer and starting to come true in a lot of other diseases is we develop molecules against these specific gas pedals to inhibit them. And that has yielded this one biomarker, one drug paradigm that work, for example, lung cancer, you have EGFR inhibitors. For breast cancers, you have HER2 inhibitors that work in patients with these specific mutations. Next slide. However, that's the cartoony one gas pedal, one person driving, right? But if you move this Mario Kart driving scenario to a real world situation, you realize it's entirely different. Most of you probably have driven a car on a real road before. It's not just, you have star power and you want to gas pedal however fast you could. Tumors cannot do that either. There's a lot of components that come into play, like if the weather is stormy that day, if there's a lot of cars around you, and then if there's a police that's watching you how fast you can go, for example, the immune system. So you end up with this complex scenario in the real world driving that the tumor really has to account for. And that might also really explain why, in some cases, the precision medicine of this one drug, one biomarker doesn't work because the scenario is complex. Next slide. And so you can think about this real world scenario where if you have all kinds of different tumors arising from different courses, there are different kind of bad guys with different, weapons, and then you have totally different sets of immune system, immune cells, and they all operate in a distinct environment. And so that's a lot of modality of data that you really need to consider when you want to find the best drug for each patient. Next slide. And to solve this, a former postdoc of mine, Rikia, who's now working in the Microsoft Siemens consortium, really come up with this idea to evaluate a personalized opportunity cost. So not just taking in the multimodality of data that compose the clinical data, the genomic data, but also the other side that we think about when we go to medicine, that we not just want to predict whether we're going to have a good outcome for immunotherapy, but we might actually want to predict simultaneously whether you have a good outcome for immunotherapy, whether you have a good outcome for chemotherapy, and evaluate your own best opportunity cost, right? As a consumer, we all like to select the best product for our own course. For medicine, we want to do that based on all the multifactorial factor in our tumor as well. So here we specifically look into immunotherapy because it's recently incorporated into a lot of standard of care and affecting a lot of standard of care. There's a lot of debate about in which case you should give immunotherapy, which case not, and which case you should give as a frontline, which case you give to recurrent disease. Next slide. The important thing to really base this study on is that now we already have a lot of sequence cohort of patients treated with immunotherapy and non-immunotherapy. So we have rigorous trained test split within the first data set, and we also want to make sure the model is generalizable whenever there's a new patients come along. So we have external data set from completely different clinical trials and other sources. I won't go into detail. Next slide. And Rikia come up with this workflow, basically pretty standard machine learning workflow, but we want to make sure it's rigorous. Next slide. And we want to make sure that eventually we can arrive at this opportunity cost model where we find patients, their probability of survival under immunotherapy, their probability of survival under standard of care therapy, non-immunotherapy, and obviously the patients that would do well in both, but more interesting, the patients that would be particularly suited to immunotherapy versus non-immunotherapy. Next slide. The other component is that we want to make sure the model that we arrive at is not just black box. So we did a bunch of testing, and in this task, we settle on XGBoost model, which is a boosted ensemble tree model. But we want to make sure it's explainable, and Shapley value is arising from this game theory framework that work well to calculate a specific player's contribution to a game outcome. And that has been generalized well to a tree model by Tree Shap, a development at University of Washington. And here we also derive specifically MSE matrix to summarize a particular contribution of a specific genetic feature or clinical feature. Next slide. And also to do machine learning and feature selection well in genomics, I think, as well as in a lot of machine learning tasks in precision medicine, we need to solve this large feature space, small sample size problem. Here, we basically derive an algorithm that leveraged a lot of a known biological prior about gene network and select gene mutations that likely arise in the same network. That won't go into detail as well. And that eventually allow us to come up with a better set of features that agree with current knowledge of how current immunotherapy work, for example. Next slide. We're able to demonstrate that when we use this kind of feature selection framework, it outperforms current standard-of-the-art feature selection, but also importantly, when you incorporate more modality of features, for example, specific gene mutation, you do outperform using key clinical metrics like just sex, ancestry, age, etc. And that incorporating specific genes mutation is very important because previously in the immunotherapy space, people have only used the sum of all mutations. Next slide. And based on the feature selection algorithm, Rikia has advised that we're able to start to see specific mutations predictive of immunotherapy outcome and non-immunotherapy outcome. So I won't go into the details of the genes, but here on the right is an example in the esophageal cancer, that you see patients with PTEN mutations actually survive better in immunotherapy. And identifying these genes are important for us not to just stratify patients, but also identify potential compound therapies that we can treat the patient with in conjunction with immunotherapy or not in conjunction with immunotherapy, for example. Next slide. Importantly, we want to make sure that the model generalizes well to patients that are outside of these treatment settings. They might be coming from a different area of the country that have different age or ancestry demographics. And here we make sure that our immunotherapy-derived model work well in external immunotherapy data sets, and non-immunotherapy model work well in the non-immunotherapy data set. But when you reverse the test for immunotherapy predict non-immunotherapy patients, they actually do not predict well, to show that these models are definitely test-specific, and we can chain them together to evaluate this opportunity cost. Next slide. And so how does this play out in a single patient level? Here on top, for example, you can see one patient with his or her specific combination of the genetic mutations and clinical matrix. You can see it sum up to a low probability of survival under immunotherapy, whereas the other patient's combination render them more susceptible to immunotherapy. Next slide. And then if we compare all these cases under the immunotherapy model and non-immunotherapy model, we can really do arrive in this opportunity cost evaluation, where at a single patient level, we're able to find the patients that are preferred for a specific immunotherapy, non-preferred for non-immunotherapy. And we really think that this will do better than the current state of the art, where we use one biomarker to bin one patient into immunotherapy-treated bin and the other into non-immunotherapy-treated bin. Next slide. And so we really hope that this, along with many other work, will move us from this one biomarker, one drug paradigm to, next slide, a more multi-model, multi-treatment AI recommendation, where we have a system that can start digesting all these information about tumors. So here we use genomics, clinical, immune data, but also in the future we can envision accompanying EHR data, accompanying histology data to come into this prediction system, not just evaluating one treatment outcome, but simultaneously evaluate multiple possible treatment to potentially provide the best recommendation to the practitioner and for the patient to decide what's the best course of therapy. Next slide. Now that's great. We're able to start moving from predicting one outcome to many. And I think many people here probably have experienced that, whether in drug discovery or I think in breeding, there's a lot of consideration about different traits of a plant, for example. The other interesting innovation that has really drawn attention recently is that when you give a lot of untrained, a lot of data to a pre-trained model, potentially you can arrive at these foundation models that understand the underlying structure of the data, and that can solve a lot of challenge in text and image has already been shown. On the next slide. And that's what we're currently really building on in, for example, the natural language foundation models that everyone know well of, like GPT, like Cloudus, but we are doing that for electronic health record. And right now we start to think about how to integrate each foundation model. Obviously, like the chronic health record is already ongoing, but if we have that for multi-omics, for longitudinal biomarker that we each get in our annual health checkup, then we can really do arrive in a really accurate understanding that in the next likely patient's visit, they might have very likely this occurrence of this disease. Or for the next course of drug, it might be best for this person with this kind of longitudinal profile to undergo this kind of therapy for preventing a disease. And that's ongoing work. Next slide. But I do want to save some time for this, I find, fascinating area of research. And I think I am more of just an amateur person playing around with this, but I think this might be the test that solves us all, right? If I want to spend most of my time just surfing, playing tennis, taking my naps, then maybe this is what we need to arrive in, is that a lot of these tasks can actually be automated. Next slide. And I think there's definitely promising development in foundation models. Like when GPT keep coming out with the next version, the performance just really, stun a lot of us. But the other thing to think about is this agentic system. So here I'm giving a not as controversial example of peer review, which is, a fundamental task that a lot of academics like us have to do. Sometimes we like it, sometimes, not as much. It just adds on a lot of work, right? But here, if you give it 0 shot, for example, as I show for GPT-4 to do a peer review, it actually do an okay job, but as many of you that has specific technical expertise, scientific expertise, like the knowledge it provide back is typically too general, typically not specific enough in the field, right? So what if we start thinking about a more agentic system where we have a more sequential reasoning, where we have more planning involved? Next slide. And so these workflow has been developed, and here I'm just showing one with the best graphics, because I guess Microsoft built this AutoGen system where you envision there are multiple agents instead of one agent solving your task at a time. And this is analogous to how a lot of us humans work in teams, that we don't just have one single person handle all the tasks, but we have specialized experts that come together as a team to best execute the task. Next slide. And so if we go back to the peer review scenario, and here actually I'm going back to a GPT 3.5 Turbo to chain together these workflow. You can imagine that we can start having different agents come together, kind of like how humans do these tasks now. So as many of you that probably have been involved in peer review knows that typically the paper first get handled by the editor, who identify 3 experts in the relevant area of expertise for them to give specific comments. And this is how the system I built actually chained together that too. So the GPT first, think about what are the 3 relevant area. And so, for example, this is actually a paper that I only had part of expertise on. That's in psychiatric genetics that use polygenic risk score that try to arrive at clinical diagnostic. And the system, or the editor of the system, is able to identify 3 specific areas. And based on these 3 specific areas, it then sequentially assume the role of each of those experts. And when it is able to do that, it actually gives much more specific insight to the paper. And obviously, you can run this many times. I didn't digest all the outputs, but this is one of the output. And this is a task that I feel like, actually the review it provided will easily, be worth more than 2-3 hours of work for me to really understand and provide critical feedback to this paper. And obviously, these systems are only evolving by the day. Next slide. Not just in the fundamental model where we have these large language models that gets better, and also in these agentic systems that people in the computation science field have continued to innovate and be able to start chaining together a lot of workflow. Here I'm showing one example. So, a lot of things that really shocked people 2 years ago, baby AGI, crew AI, are considered really outdated now, right? And the React framework, which chains together the reasoning and chain of thought. So for example, priming the large language model to think about solving the problem by specific chain of thoughts of reasoning that has worked well, tying together with action, now has grown into using a compiler-like system. And how they do this is that they will break down the task into multiple subtasks and find out which ones need to be done sequentially, which one need to be done parallelly, and then have executor using each different tools to start putting all these together. So this is arriving in a state that I think is really starting to become like how human teams work together. And perhaps this will be our future kind of operating system, that instead of each of us going to operating system, click on each of the apps, the LLM is going to be the one that's mostly doing it, and humans go to it with an important task. Or next slide. I think or the other side we have to really think about is how much and where do humans really add unique value. And in biomedicine, I think we encounter a lot of these fundamental challenges, like a lot of other fields have to think about. A lot of the big questions become philosophical and moral. So errors, obviously there's a lot of hallucinations, just like humans will make the errors, right? But what's acceptable? Is it just the same error rate as human? Is it tenfold lower error rate than humans? In which use case? And who will be responsible for those misdiagnoses, for example, for those mistreatment, for example? What will be the role of human in these systems? I think we can already start to see a glimpse in some fields of pathology where AI has outperformed average or even the best humans sometimes, that the pathologist is required still to make the final destination of a report. So we have that final stamp on that. And I feel like a lot of human roles might go into these regulatory side of things to make sure everything works right. And lastly, I think it's very important for us to have public trust and communication of these systems, how they work, for example, to help patients think about what the AI system output as a recommended treatment and how do they reconcile that with what a doctor think and how they think for themselves. Next slide. So with that, I'd like to thank all of you for your attention, the URLs to my lab, and welcome any questions you might have. Thank you.